Uh, Today I'm going to talking about the fourth chapter of the AS Edexcel Biology. So here subchapter gene pool and genetic diversity. First of all, I'm telling about what we are planning to study here. What is the population? First of all, we have to understand what is the population and gene pool and allele frequency and what is the Hardy-Weinberg equation, what are the conditions what we have to follow when you are using the Hardy-Weinberg equation, something like that. You have to study these facts under that subchapter. So population, what is the population? Okay, actually according to this is my short note actually. So here you can see clearly. Okay, you can't clear. Population, it's like a groups of same species. That's like a collection. What is it? In your textbook there is included proper definition for that, but there is a specific word, same species. The collection of groups in the same species we are called as the population. Okay, so population means actually, it's an example when you get dogs. Okay, dogs are so many variation. Because of that variation they are categorized into so many groups. Okay, these groups all together, when we considering in a particular area, particular habitat, that is we called as the population. For the population actually there is included the interactions also, group wise interaction are there, and also with the habitat interaction, living non-living interaction. That's why we are telling in the population, when you are studying physical elements and biological elements, we have to identify, we have to understand. Physical element means behavioral patterns. Okay, biological elements mean respiration, digestion, something like that. Okay, so guys, this is the basic definition of the population. And when you are considering of the niche, what is the meaning of the niche? That's like what is the role of that particular species to that habitat. Okay, something like that. And what is the gene pool? Gene, first of all, what is the gene? Gene means actually it's like a specific base arrangement of a part of a DNA, that's a chromosome. So that is a gene. No? Gene responsible for a particular character. Okay, so because of that reason, from one generation to another generation, the particular characters has to be transported through the genes, through the DNA, to another generations also. So in a population, all the genes are collections. Gene collection in a same species we are called as in a particular time also we called as the gene pool. Okay, if there is any mutations of the genes, like specific arrangement can be changed, that's a mutation. Okay, that is can be also transport one generation to another generation, then the particular character can be modified. Okay, these are those theories you have to apply for the allele frequencies and also the Hardy-Weinberg equation. Next I'm going to talking with you what is the allele frequency. Okay, allele means actually when we consider the diploid organism, heterozygote, consider dominance and recessive combination is the heterozygote name. So dominant one is if it is capital A, the recessive one is simple A. Altogether capital A and simple A we called as the heterozygote diploid organism. Okay, when you are considering about during the meiosis, what happened, the diploid one becomes haploid now. Okay, so here capital A and simple A will be separated out. Okay, So when we consider heterozygote diploid organisms 100 in a population, in a particular habitat, so what happened after that? Simple A and the capital A divided. Then capital A also 100, simple A also 100. All together there is 100 organism. But allele wise there is capital A 100, simple A 100. Then all together when we consider the whole number of alleles in that population is 200. Okay, 200. In your examination questions they ask what is the capital A allele frequency in that particular habitat. Then what you have to do is the A number of alleles of A divided by total number of alleles. Okay, above considered, according to the above considered example, capital A is also 100. Capital A 100 means altogether alleles, the whole number of alleles are 200. 100 divided by 200, that's a 0.5. Okay, that means always allele frequency cannot be larger than 1. It's a ratio. So something like this way. So here 100 heterozygote diploid organism. This is heterozygote. This is the presentation of heterozygote. Then A allele, simple A allele. This is 100, but 100 alleles, 100 alleles. Okay. Then A allele frequency is 0.5. Can you understand? So that's the way to find out the allele frequency, normal method. Okay. Then after that, there were simple equation to represent this. We normally call as the dominant allele frequency plus recessive allele frequency is always 1 in the population. Okay. Population analysis, we are taking so one calculation. There is a simple equation P plus Q. What is the P plus Q? P means dominant allele frequency, Q means recessive allele frequency. In a population, P plus Q equal 1. Okay, so now this is a small equation, yeah, that's very simple. But when we apply this is very hard. Why is that? Normally genotypes, when we are considering genotype, we can consider they are the heterozygote people, heterozygote organisms. Those are the dominant homozygous. But when you are just thinking about the phenotype, we could not be able to separate out them easily. Can you understand what I am saying? So here guys, consider tallest people, capital T, capital T, capital T, simple T. When I consider that one, heterozygote one also showing the phenotype is tall. Dominant homozygous one also showing phenotype is tall now, but recessive one is simple T, that is tall. You can understand that's with the external appearance. To the external appearance you can understand that. Okay, but in a population we could not be able to distinguish heterozygote one, the dominant homozygous one, very easily. So because of that reason they are telling it's very hard to use the P plus Q equal 1 equation for a particular population to find out the frequency of the dominant homozygous one and heterozygous organism. Okay, so because of that reason the scientists understood there should be another equation for that. Okay, that's the Hardy-Weinberg equation. So what is the Hardy-Weinberg equation? That's a solution to calculate the frequencies of the dominant and heterozygote organism, dominant homozygous and heterozygote organisms. So here for the Hardy-Weinberg equation you can apply under some conditions. There are conditions you have to follow to apply the Hardy-Weinberg equation. Okay, so what are the conditions? Normally, first of all, in your textbook there is included the there is the with the informations for the information that's included the conditions they explained also. Okay, go through that one. This is a small explanation. So first one, definitely population should be stable. That means when we consider the population in a particular habitat, when we are doing that analysis, there should not be any immigrations or any migration. There should not be included or happen. Can you understand? So, and also population, that's inside the population from one generation to another generation, particular allele frequency should be stable, and they are not inside the population and inside the particular habitat. Random mating could not be happen. Okay, and also no evolutionary changes, because if there is any evolutionary changes happen, the allele frequency also can be changed. So the allele frequency is not stable, then we cannot use the Hardy-Weinberg equation for that particular population. Okay, depending on the above conditions, assuming that are okay with the population, so then we can use the Hardy-Weinberg equation. Okay, from the simple equation that is deviated into the little bit complex equation, p square plus 2pq plus q square equal 1. That is the Hardy-Weinberg equilibrium equation. Okay, p squared here, p squared 2pq plus q squared equal 1. What is the p squared? What is the p squared? P squared means the frequency of homozygote dominant organism, diploid organism. 2pq means frequency of the heterozygous diploid organism, and q squared means frequency of the homozygous recessive diploid organism equal 1. Okay, so guys, inside the population, the recessive homozygous recessive one, if that's included, definitely we can identify. No, in your examination questions, basically they are given the frequency of the recessive homozygous organism. Something like, consider there is a one example for your Edexcel book, page number 278 page. There's an example here. There's included all of you know about the albino, that disease. Actually albino is that trait is represented by the homozygous recessive allele. No, the heterozygous one also not expressed. It's like hiding, and also dominant zygous is not express the albino. So albino trait, for the albino trait they are telling, test on a sample North American showed that frequency of albinos in the population was 1 in 20,000. 1 in 20,000. That means the North American showed that the frequency of albinos, when they are telling about when they are telling about the frequency of albinos, means definitely 1 over 2,000 allele frequencies related to the albino trait. This tells us albino trait means for the homozygous recessive people. This tells us that frequency q squared of the homozygous recessive trait is 1 over 20,000. Okay, if you know that q squared is equal 1 over 20,000, okay, q squared, because they are telling about the Simple note that show the frequency of albinos. Here albinos means diploid one, because from a one allele you can't express a trait. To express a trait, express a character, there should be two alleles. No, inside the population, when they are telling that trait or whatever disease expressed by 1 over 20,000 means actually that could be a diploid organism. Okay, so Q squared is equal to 1 over 20,000. So Q squared is 1 over 20,000. Then you can find out the root value of the Q squared. Then if it is X, using the previous simple equation P plus Q equal 1, you can find out P value. Then after that you can find the dominant homozygous allele frequency P squared and also heterozygous allele frequency by using 2pq. So that's a small equation, that's a small calculation, and you give it in your examination small calculation, sometimes they will give to resolve and to find out what is the homozygous dominant allele frequency and what is the heterozygous allele frequency. So then it's easy for you to find out by using this method. Okay, so if you have any question, if you have any problem, you have to use my Facebook, and also you can put in box me a message, then I can help you. So guys, later on I will decide to come back to another lessons also. So until that, goodbye for all of you.